Welcome back, everybody. Today we're doing a deep dive into a world where DNA, well, it's not just a straight ladder anymore.
That's right.
We're going to be talking about how the shape of DNA impacts our ability to learn and form memories.
That's a great way to put it.
Buckle up, because this is going to be a really fascinating one.
It is.
We are diving specifically into an April 2024 Journal of Neuroscience article called DNA G-Quadruplex is a transcriptional control device that regulates memory. So what's the big takeaway here?
Well, I think the biggest takeaway is this research is focused on something called G-Quadruplex DNA, or G4-DNA for short.
Okay.
And this is a really interesting structure that's been found to be increasingly important.
G4-DNA? That sounds pretty intense.
It does.
Is this something out of a science fiction movie?
It might sound a little futuristic, but it's very real. And it's formed when guanine bases in DNA come together in a very specific way.
Okay.
And it's kind of like if you imagine folding a flat piece of paper into a complex origami shape. That's kind of what's happening with G4-DNA.
So it's not just a straight ladder anymore.
Nope.
We're talking DNA origami.
Exactly.
I'm intrigued, but how does this all tie back to our brains and our memories?
Well, that's exactly what these researchers wanted to find out. And so they designed experiments with mice where they trained the mice to associate a specific sound with a mild electric shock.
The classic fear conditioning experiment.
Yeah.
I'm guessing these mice learned pretty quickly.
They did. And here's the really interesting part. As the mice were learning, the amount of G4-DNA in their neurons actually increased.
Oh.
And they used a technique called G4-seq to pinpoint the location of G4-DNA in the brain.
Hold on, so you're telling me that learning is somehow triggering the formation of this G4-DNA?
Yeah.
That's pretty wild.
It really is, but it gets even more interesting.
Okay.
Because they also identified this enzyme called DHX36, which acts like a G4-DNA unwinder.
Okay.
So it basically smooths those origami folds back into a more typical DNA shape.
So we've got G4-DNA forming during learning, and then we have DHX36 coming in and potentially like undoing those formations.
Right.
I have a feeling this is where things start to get really complex.
Yeah, you're right. They wanted to see what would happen if they tipped the scales a little bit.
Okay.
So they used a special technique to reduce the amount of DHX36 in the mouse's brains.
So with less DHX36 around, more G4-DNA would be able to stick around.
Exactly.
That boosts their memory?
That's what you might expect, right?
Yeah.
But here's where things take a really unexpected turn. The mice with reduced DHX36 and more G4-DNA actually had a harder time forming and holding on to certain types of memories.
Wait, so more G4-DNA actually made their memories worse?
Yeah.
I'm officially confused. I thought we were building to G4-DNA being a good thing for memory.
It's not quite as simple as that, and this is where this concept of G4-DNA acting as a switch for gene expression comes into play.
Okay, time for a quick refresher. Genes are sections of DNA that provide instructions for building proteins, and proteins carry out all sorts of essential functions in our cells, right?
Exactly.
So how does G4-DNA fit into that?
Well, imagine G4-DNA as a dimmer switch on those instructions.
Okay.
Sometimes when G4-DNA forms, it can actually dim the signal, effectively turning certain genes off. But in other cases, and we're still trying to understand why G4-DNA can actually amplify the signal, making those genes more active.
So G4-DNA can flip the switch either way.
Right.
That's a lot of power for a tiny structure.
Absolutely.
Yeah.
And in this study, the researchers found that G4-DNA seems to be regulating genes that are critical for both forming memories and for synaptic plasticity.
Synaptic plasticity. Remind me what that is again.
It's the ability of connections between neurons to strengthen or weaken over time.
Okay.
Think of it like the brain's way of adapting and rewiring itself based on our experiences.
So G4-DNA is influencing the very processes that allow our brains to change and learn. This is starting to feel like a real-life version of Inception, all these layers within layers inside our brains.
Right.
But we can't forget about RNA.
Right.
It's DNA's close cousin and plays a vital role in converting those genetic instructions into proteins.
So did the researchers look into that?
Yeah, they did, and they used cutting-edge techniques to study how the presence of G4-DNA affects both the amount of RNA produced from certain genes and the way that RNA is processed.
So it's not just about turning genes on or off, but also controlling the volume and fine-tuning the output.
That's a great way to put it.
Wow.
And the more we learn, the more we realize just how multifaceted the relationship between G4-DNA, RNA, and gene expression really is.
To really wrap our heads around this, let's zero in on a specific example. The researchers focused on two particular genes, GFN and SHL1.
Okay.
What makes these stand out?
These genes are particularly interesting because they play key roles in how neurons connect and communicate with each other, essentially impacting the wiring and signaling of the brain. So by manipulating G4-DNA at these specific genes, the researchers could directly affect how neurons talk to each other.
That's right.
What did they find?
Well, they discovered that manipulating G4-DNA at GCN and GHL1 had different effects on memory formation in mice.
Wow, that's a level of precision I wouldn't have imagined. It's incredible to think that something as seemingly simple as the shape of DNA can have such profound effects on something as complex as memory.
It really underscores the incredible complexity of the brain and the fact that we're only just beginning to scratch the surface of how it all works.
I'm already questioning everything I thought I knew about DNA.
That's the beauty of scientific discovery.
Yeah.
It challenges our assumptions and opens up whole new worlds of understanding.
Speaking of new worlds, before we get too lost in the details, I think it's time to zoom out and consider the bigger picture.
Okay.
What are the implications of all of this? What does this research mean for us?
Well, this study provides some of the strongest evidence to date that dynamic changes in DNA structure, specifically the formation of G4-DNA, play a crucial role in how our brains learn and form memories.
So it's not just about the sequence of our DNA, the classic A, T, C, and Gs we learned about in school.
Right.
It's also about how those building blocks are physically arranged in space.
Exactly. It adds a whole new layer of complexity to how we think about genetic regulation. And this has huge implications for how we approach memory disorders.
That was my next question. Could this research lead to new treatments for conditions like Alzheimer's or dementia?
It's definitely a possibility. If we can understand how G4-DNA formation and regulation contribute to memory problems, we might be able to develop therapies that target those specific mechanisms.
Imagine being able to manipulate the shape of DNA to improve memory function. That would be revolutionary.
It's a long road from basic research to clinical applications. But this study opens up some incredibly exciting avenues for exploration.
This deep dive is giving me hope for the future of neuroscience.
Me too.
But before we get too carried away, you mentioned earlier that we're just scratching the surface.
Yeah.
What are some of the big unanswered questions about G4-DNA in the brain?
Well, for one, how does G4-DNA formation differ between different types of neurons?
Okay.
And what are the long-term effects of manipulating G4-DNA?
Right.
Could there be unforeseen side effects?
So many questions.
Yeah.
But that's what makes science so captivating, right?
Absolutely.
It's a never-ending quest for knowledge and understanding.
It really is. Each discovery, like the one we've been discussing today, brings us one step closer to unlocking the mysteries of the brain.
Before we wrap up this part of our deep dive, I want to make sure our listeners have a clear takeaway from all of this. Can you sum up the key findings in a way that's easy to digest?
Certainly. The researchers demonstrated that G4-DNA accumulates in the brains of mice during the formation of fear memories. They also showed that reducing the levels of the DHX36 enzyme, which unwinds G4-DNA, actually impaired memory in these mice. This strongly suggests that the dynamic formation and unwinding of G4-DNA is crucial for memory processes.
So it's like a delicate dance inside our cells.
Yeah.
Too much G4-DNA or too little and things get out of sync.
That's a great way to put it. And it highlights the fact that the shape of DNA can be just as important as its sequence when it comes to brain function.
It's mind-boggling to think that something so tiny and fundamental can have such a big impact on our ability to learn, remember, and ultimately who we are.
That's the wonder of science. It constantly reminds us of the interconnectedness of everything from the smallest molecules to the most complex thoughts and emotions.
Speaking of thoughts and emotions, I'm feeling a wave of curiosity washing over me. I need to know more.
That's the spirit. And the good news is there's a whole world of research out there waiting to be explored. We'll delve deeper into some of that in the next part of our deep dive. Stay tuned.
All right. I'm ready to go even deeper down this G4-DNA rabbit hole. Where do we go from here?
Well, remember how we talked about G4-DNA acting like a switch for gene expression?
Yeah, it can turn genes on or off, like a dimmer switch.
Exactly. And the researchers in this study found that G4-DNA seems to be particularly involved in regulating genes that are crucial for both forming memories and for synaptic plasticity.
Hold on, can you remind me what synaptic plasticity is again?
Think of it like this. Every time you learn something new, your brain is literally rewiring itself. Synaptic plasticity refers to the ability of the connections between neurons called synapses to strengthen or weaken over time. This is essentially how our brains adapt and store information.
Okay, I'm starting to see the connection here. So G4-DNA is somehow influencing how those connections between neurons change, ultimately affecting how memories are formed and retained.
That's the idea. It's like G4-DNA is playing a role in orchestrating the very process of learning and memory at a fundamental level.
It's incredible to think that something as seemingly simple as the shape of DNA can have such a profound impact on something as complex as memory. It's making me wonder, what are the implications of this research for us, for humans? I mean, these experiments were done in mice, right?
Right. But mice and humans share a lot of similarities when it comes to basic biological processes, including how our brains learn and form memories. So while we can't directly extrapolate these findings to humans just yet, it definitely opens up some fascinating possibilities.
So what you're saying is this research could potentially revolutionize how we think about memory and learning in humans.
Absolutely. Imagine if we could develop targeted therapies that influence G4-DNA formation and regulation. It could lead to new treatments for memory disorders like Alzheimer's disease, or even help people enhance their cognitive abilities.
Now that's a future I can get excited about. But let's be realistic for a moment. There's still a lot we don't know about G4-DNA and its role in the brain, right?
Oh, absolutely. This study is really just the tip of the iceberg. There's a whole world of questions still to be explored.
Like what? Give me some examples.
For starters, we need to understand how G4-DNA formation varies across different types of neurons. Do certain neurons rely more heavily on G4-DNA for memory formation than others? And what are the long-term consequences of manipulating G4-DNA? Could there be unforeseen side effects?
Wow, so many questions. I'm starting to realize that we're not just dealing with a single memory molecule here. It's more like a complex ecosystem with G4-DNA playing a key role in shaping the landscape. It's almost like those nature documentaries where they zoom into a seemingly simple ecosystem, like a drop of water, and suddenly you realize there's an entire universe of activity happening right before your eyes.
That's a great analogy.
Yeah.
And it highlights just how much more there is to discover. But I think it's also important to acknowledge what a major breakthrough this study represents. It's a significant step forward in our understanding of how memory works at a molecular level.
I agree. This research really makes you appreciate the incredible complexity and elegance of the brain. To think that something as tiny and seemingly insignificant as the shape of DNA can have such a profound impact on who we are and how we experience the world is truly awe-inspiring.
It really is. And it's a reminder that even the smallest discoveries can have ripple effects that extend far beyond the lab.
So where do we go from here? How can we connect this groundbreaking research back to the listener, to their everyday life?
Well, I think the biggest takeaway is that our brains are incredibly dynamic and adaptable. Even at the level of our DNA, there's constant change and adaptation happening. And while we're only just beginning to understand the intricacies of G4-DNA and its role in memory, it's clear that this research has the potential to transform how we approach learning memory and even our own identities.
That's a powerful thought. It makes you wonder, what if we could harness the power of G4-DNA to enhance our cognitive abilities or even prevent memory loss as we age?
Those are some of the big questions that researchers are now grappling with. And as we delve deeper into this fascinating world of DNA origami, I have a feeling we're going to uncover even more surprising and profound insights about ourselves.
This deep dive has definitely sparked my curiosity. What else can you tell us about the potential applications of this research?
One area that's particularly exciting is the development of new drugs and therapies for memory disorders. If we can understand how to precisely target and manipulate G4-DNA formation, we might be able to develop treatments that are more effective and have fewer side effects than current options. Imagine a drug that could help people with Alzheimer's disease regain lost memories or prevent further cognitive decline.
That would be incredible. It's almost like science fiction becoming reality. But I'm also curious about the ethical implications. If we're talking about manipulating something as fundamental as DNA, we need to proceed with caution, right?
Absolutely. As with any powerful new technology, there are always ethical considerations. It's crucial that we have open and honest conversations about the potential benefits and risks of manipulating G4-DNA and ensure that any applications are developed and used responsibly.
I completely agree. This research is incredibly promising, but it's important to remember that we're still in the early stages of understanding. We need to balance our excitement with a healthy dose of caution and responsibility.
Well said. And that brings us to another fascinating aspect of this research: the potential for enhancing cognitive function in healthy individuals.
You mean like a smart pill that boosts memory and learning?
It's a possibility, but it's also a topic that raises a lot of ethical questions. Should we be trying to enhance our cognitive abilities beyond what's considered normal? What are the potential societal implications of such a technology?
These are questions that we as a society will need to grapple with as this research progresses. It's a reminder that scientific advancements don't happen in a vacuum. They always have broader ethical and societal implications that we need to consider carefully.
You're absolutely right. And that's why it's so important to have open dialogues about these issues involving not just scientists, but also ethicists, policymakers, and the public at large. We need to ensure that these powerful new technologies are used in ways that benefit humanity as a whole.
Well said. This deep dive has certainly given us a lot to think about, hasn't it? We've gone from the intricate world of DNA origami to the potential for revolutionizing how we approach memory learning and even the very essence of who we are. It's been an incredible journey so far.
It really has. And as we wrap up this part of our deep dive, I think it's safe to say that the future of G4-DNA research is incredibly exciting. I, for one, can't wait to see what groundbreaking discoveries lie ahead.
Me too. But before we get too carried away with the future, let's take a moment to recap some of the key insights from this part of our discussion. What are the main takeaways you'd like listeners to remember?
Well, I think the most important point is that G4-DNA appears to play a crucial role in regulating the genes involved in memory formation and synaptic plasticity. This means that the shape of DNA, not just its sequence, can have a profound impact on how our brains learn and adapt.
Right. And we've also learned that manipulating G4-DNA can both impair and enhance memory in mice, suggesting that there's a delicate balance at play. This opens up some incredible possibilities for developing new treatments for memory disorders, but also raises ethical questions about the potential for cognitive enhancement in healthy individuals.
Exactly. There's a lot more research to be done, but it's clear that G4-DNA is a powerful force to be reckoned with in the world of neuroscience.
I couldn't agree more. This deep dive has been an eye-opening experience. But before we wrap up entirely, I want to give our listeners a taste of what's to come in the final part of our exploration.
I think it's time to shift our focus from the microscopic world of DNA to the vast and complex landscape of the human brain. Let's delve into the specific regions of the brain involved in memory and explore how G4-DNA might be influencing these regions in ways we're only just beginning to understand.
Sounds fascinating. I can't wait to unravel these mysteries. But for now, we'll leave our listeners hanging on that cliffhanger. Be sure to join us for the final part of our deep dive, where we'll journey into the depths of the human brain and uncover even more secrets about the intricate dance between G4-DNA and memory. Welcome back for the final part of our deep dive into G4-DNA and memory. We've talked about DNA origami and how it could impact memory disorders and learning, and now I think it's time to really dive into the human brain.
All right, let's do it.
I'm ready.
Okay, so let's start with the hippocampus. This is a seahorse-shaped structure deep within the brain.
Okay.
And it's absolutely essential for forming new memories, especially those related to facts and events. Think of it like the brain's memory filing cabinet.
So if the hippocampus is damaged, you would have trouble forming new memories.
Exactly. And that's why damage to the hippocampus is often seen in people with Alzheimer's disease, which is characterized by severe memory loss.
I see. So how does G4-DNA fit into all of this?
Well, researchers are now finding that G4-DNA formation might be particularly active in the hippocampus.
Yeah.
Remember how we talked about G4-DNA acting like a dimmer switch for gene expression?
Yeah.
It seems that this process might be really crucial for the hippocampus to do its job effectively.
So if there's a problem with G4-DNA regulation in the hippocampus, it could disrupt memory formation.
That's the hypothesis.
Yeah.
And it's an area of intense research right now.
Yeah.
Scientists are really trying to understand exactly how G4-DNA influences gene expression in the hippocampus.
Okay.
And whether manipulating G4-DNA could potentially protect against memory loss or even restore lost memories.
That's amazing. It's almost like we're on the verge of unlocking a secret toad within our own brains.
Yeah.
But the hippocampus isn't the only brain region involved in memory, is it?
Oh, absolutely not.
Okay.
Another key player is the amygdala. It's a small almond-shaped structure that plays a central role in processing emotions, particularly fear and anxiety.
Okay, so the amygdala is all about our emotional responses. How does memory tie into that?
Well, the amygdala is really important for forming what are called emotional memories. Have you ever noticed how certain smells or sounds can trigger vivid memories, often accompanied by strong emotions?
You mean like Proust's Madeleine? How the taste of a simple cookie evoked a flood of childhood memories?
Exactly. That's the amygdala at work. It's deeply involved in linking memories with emotions. And what's really fascinating is that researchers are now investigating whether G4-DNA might also play a role in this process.
Hold on, so G4-DNA isn't just about forming memories in general. It might actually influence how those memories are emotionally charged.
That's a possibility that's being actively explored right now.
Hmm.
Imagine if we could understand how G4-DNA regulates gene expression in the amygdala, and how this in turn affects emotional memory formation.
Yeah.
It could open up entirely new avenues for treating conditions like PTSD, where traumatic memories are often accompanied by intense fear and anxiety.
Wow, this research just keeps getting more and more intriguing. It's amazing to think that something as tiny and fundamental as the shape of DNA could have such a profound impact on the way we experience and remember the world.
It really is. And it's a reminder that the human brain is an incredibly complex and interconnected organ. We're only just beginning to scratch the surface of how it all works. But with each new discovery, we're gaining a deeper appreciation for the intricate dance between our genes, our experiences, and our memories.
I totally agree. This deep dive has been a real eye-opener. We've gone from DNA origami to the potential for revolutionizing how we think about memory learning and even the essence of who we are.
Yeah, and as we wrap up our exploration, I want to leave you with a thought to ponder. We often think of our DNA as a fixed blueprint, a set of instructions passed down from our ancestors.
Mm.
But what if it's more dynamic than we ever imagined? What if the very shape of our DNA and its influence on gene expression is constantly changing in response to our experiences, shaping our brains and our memories in ways we're only just beginning to understand?
That's a fascinating and somewhat humbling thought. It makes you realize that we're not just passive recipients of our genetic heritage, we're actively shaping our own destinies right down to the molecular level. And with these incredible advances in research, we're gaining the tools to understand and potentially even influence these processes in ways that could benefit humanity as a whole.
Well said. And I think that's a perfect note to end on.
Hmm.
This deep dive has been a journey of discovery exploring the intricate world of G4-DNA and its impact on memory. We've uncovered some truly amazing insights, and I hope it sparked your curiosity to learn even more.
I know mine has. A huge thank you to our expert for guiding us through this complex and fascinating landscape. And to our listeners, thank you for joining us on this incredible adventure into the depths of the human brain. Until next time, keep exploring, keep questioning, and keep those synapses firing.